For the second half of the week 2 lecture on advanced imaging, I'm going to tell you about some of the super resolution techniques in light microscopy. Then I'll go on to just a quick little bit about image analysis, and then a few slides at the end about electron microscopy. So, super resolution. Way back at the start of the first lecture, I was talking about resolution. And I told you about the theoretical maximum resolution of light and electron microscopes. And at that point, I said that a light microscope, and I mean a big research light microscope equipped with the very best objective lenses, the ones with the highest numerical apertures, could resolve things as small as 200 nanometers or thereabouts. Well, super resolution techniques have come along in the last 10 years, and these are techniques that let us break this theoretical maximum resolving limit. So we can go down with some of these super resolution techniques to maybe 50 nanometers, more commonly 100 to 150 nanometers. And they're generally done using a little bit of trickery, whether that be in the microscope or in the software. So the first one I want to talk about is one that we have available at Oxford Brookes on our confocal microscopes, and this is called the Airyscan technique. Fluorescent signal from an excited point is detected by an array of 32 detectors. So that's different from in a standard confocal microscope where the excitation point sends up a signal which is detected after passing through a pinhole into a single detector. So Airyscan uses 32 detectors, sums the signal from all detectors, and produces an image that's brighter and cleaner than that from a normal or conventional confocal microscope. So instead of rejecting some of the light, as in a conventional pinhole-based system, all of the light is collected, and then the diffracted light is reassigned back to the point where it came from, which gives that nice, bright, clear image that I'm talking about. If you look at the images down here in the bottom left, this is a cell. It's got some cytoskeletal elements marked in green and various other things marked in purple and red. I don't even know what the other structures are in this picture. By the way, there could be an image to the left of this over here that showed regular epifluorescence. If you were seeing the view through a regular fluorescence microscope, you would just see a blur. You would see some green and some purple and red, but you wouldn't really be able to make out what the structures were. So this image here shows the confocal microscope picture, and I mean, it's generally a nice, clear, really, really amazing image. It's really, really very beautiful. This one here is blown up quite a lot, which is why maybe it looks a little bit blurry. But if we make the same sort of image of the slide, but we use the Airyscan technique, you can see how cleared up it is, how we can start to resolve those very, very nice, fine actin microfilaments. If that's what they are, they might be microtubules, I'm not sure, but they are clean and beautiful. So our hope with Airyscan imaging is that we'll be able to zoom up and we'll be able to look at very, very small things within cells, and we'll be able to resolve them better so that we can see, for example, if they're interacting with each other. I don't even know if you've noticed that this is a movie that's playing over here in the corner to a GFP. So this is tubulin that's been fused to GFP, that forms the microtubules. And if you study the movie really carefully, you can actually see the beautiful detail of the microtubules growing. Here's one up here that's growing during the duration of that movie. We can see the tip growth going on. You can see the remodeling that's happening in the microtubule cytoskeleton. So really rather beautiful. We move on to the techniques that are called super resolution. They're truly super resolution techniques. The Zeiss Airyscan technique that I just told you about is what I call near super resolution. It gives you a bit of an increase in resolution based on the confocal, but whether it's really, really true, whether we're getting down to that 100 nanometer range, I'm not sure. The most famous and the most useful, as far as I'm concerned, of the super resolution techniques is this one that's called stimulated emission depletion. Look at the picture here. From a confocal microscope, you use the laser to excite a region. And this region here contains a little string. I don't know what it is. Let's pretend it's one of those microtubules that we were just looking at in the movie on the last slide. There's all sorts of little dots along the microtubule, and those are the tubulin molecules that have been linked with maybe GFP. So the laser excites And you can see that it excites in a region that is intense in the center, but it has some sort of width here. And that causes low-level excitation all around that microtubule. That tends to excite the GFP, whatever it is. It could be RFP, it's colored red there. It excites it in a region around where the laser was targeted, and you get this fluorescence that's sort of generalized to an area. It's lighting up all of the fluorochromes in that area there. STED goes one step further. It uses the laser, and it excites a region just like this here, and you can see that region in the center of this donut. But what it does is it fires a ring of laser light in a different color, which bleaches all of the fluorochromes that have been excited from out at the edge here. So rather than having a whole lot of fluorescent molecules like here, you've got one or two excited right in the very center of that excitation region, and you've got a bunch that were excited but then depleted by the so-called depletion laser. And that means that you've got a nice sharp view of this one line here, which again might be a microtubule or something like that. You can compare confocal images of particles that have been excited with STED images of particles that have been excited. Very same magnification, and look at the difference. Look at the resolving power of this STED system, how beautiful and clear those little dots are. And what's happened there is that the width, black line to black line, of these little dots here has been reduced. This is a scale of resolution here from 0, which would be a vanishingly small point, to the width of the dot that you image using both systems. So the red line indicates the width of these dots in STED microscopy, and the black peak here at halfway, which is called the full width half maximum, indicates the diameter of these spots here. So there's a substantial increase in resolution by using the depletion laser in a STED microscope setup. And here's another picture here, a real-world picture. What they did was they imaged this nucleus at very high magnification using standard confocal microscopy, and what's indicated here are the nuclear pores. The nuclear pore complexes have been marked with two different fluorescent proteins, a green one in the center of the complex and red indicating structures around the outside of the nuclear pore. This would have been a remarkably clear image compared to fluorescence microscopy. At lower magnification, you would be able to see that these were dots, but you wouldn't be able to resolve any structure inside of the dots. Look here when we use STED, so these are nuclear pores in frog cells. You can see the structure of the nuclear pores, green fluorochrome in the center, and all of these red structures out the outside. So that was it for super resolution. There are other super resolution techniques that I could talk about, but I'm conscious of time and I want to move on a little bit here. The one thing that we've got to talk about when we talk about microscopy is image analysis. You can sit in your little dark room all day long making beautiful pictures. I've been showing lots of pictures of living cells. You make time series movies by repeatedly collecting images of the same plane of focus like this. So you might set up and you might say, well, I want to image this cell for 1 minute, which is how I did those FRAP experiments that I showed you earlier. Or it might be a slower process like cell division, and you might say, image this field of view for 30 minutes, make, for example, 1 image every 15 seconds. So the microscope does that. It scans the field of view, waits 15 seconds, scans the field of view again, and does that for 30 minutes. So that's 4 images per minute for 30 minutes, something like 120 images, and you put all of those 120 images together, speed them up, and that's how you make a video, a time lapse, as it were. So you can generate a huge number of images. In some of our FRAP experiments, we will collect 300 images as fast as we can, 1 image after the other, 300 images stored into the memory of the microscope. What do you do with all that data? Remember, another thing you do with confocal microscopy is you do the optical sectioning, the so-called Z series, where you step down through a relatively three-dimensional specimen. Well, you can step down through and make 50 or 60 or 100 different planes of focus, and you can do that in several different channels, a green channel, a blue channel, a red channel, for example. So that would be let's say 50 Z steps times 3 channels would be 150 images. You have these absolutely gigantic data sets. So this is where image analysis comes in. Now, image analysis is basically opening a digital image file on a computer and making measurements. There are lots and lots of commercial image analysis software packages available. Some of them cost thousands or tens of thousands of pounds, depending on what they're made to do. The microscope systems that I've been talking about, both the confocal and the electron microscope systems that I will introduce in a bit, all come with their own software that's pretty capable. The Zeiss confocal software that we use has all sorts of inbuilt image analysis software. It will analyze FRAP experiments and calculate mobility. It will analyze the amount of fret that happens if you're doing a fret experiment, for example. It will allow you to make measurements in images. Some of the most very basic image analysis is making measurements of cells. How wide are they? How long are they? How fast do they move? Things like that. So I've got to take you back, and I'm going to tell you a little story about the sort of history of image analysis in the digital age, let's say. If you want to analyze images, you don't need to go out and spend tens of thousands of pounds necessarily. You can come into the confocal lab, collect a bunch of data, go away, and analyze it on your own computer. And the reason you can analyze it on your own computer without specialist software is because there's a free software available. And the free software is called, it's written right there, ImageJ. I've said down here, ImageJ, presenting the world's most boring looking website, and is it ever? And I mean, I think on purpose. I'll tell you the story of ImageJ. But anyway, let me set the scene a little bit here. Consider a black and white image taken of a group of cells. I don't have it. What I should do is get these couple of images and put them up on the screen. I'm thinking of them. I hope that helps you. But a black and white image taken from above of a bunch of cells growing on a petri dish. Sitting beside on your computer screen, another black and white image of the surface of the moon with a whole bunch of craters. You've got those two pictures. They actually look, to the untrained eye, relatively similar. Black and white image with a bunch of little circles in it. You want to make measurements. You want to know what the diameter of the cells is, let's say. Or you want to know what the diameter of the craters on the moon is. Well, clearly we're working with two completely different scales of magnification here. So any image analysis software needs to know what magnification was used to make the original image. Modern image analysis software packages read the data files that you put into them, and the data file stores in its header, in its information file, it stores all of the information. In a confocal system, it says which objective you've used, how much zoom magnification there was, and all sorts of things like that. So the software package that you use to open your image knows exactly what magnification it's looking at. If you started measuring the picture of the moon taken with your telescope, it would know that a little line you draw represents miles or kilometers. Whereas if you're looking at cells, it knows that the little line you draw and say measure represents micrometers, let's say, something like that. So image analysis packages are very, very clever these days. But I told you I was going to tell you the story about ImageJ. This goes back to the 1980s. With the advent of personal computing, there was a fellow named Wayne Rasbad who worked at the National Institute of Health in America. And he was a computer expert, and he was noticing that these new Macintosh computers, the Apple Macintosh computer, were very good at opening up images. And he had a requirement for making images of the things they were seeing in their images. They were, what was he? He was looking at X-rays or something like that, and he wanted to make measurements in X-rays. And he said, what would be great is if I could draw a line in the X-ray image, and it would tell me how long that line was in real life. And so this was the genesis of this program. So he made the program and he wrote it in what is nowadays considered very old-timey computer programming language, and he got it working, and colleagues of his saw over his shoulder what he was doing on his computer, and they said, hey, can I get a copy of that program. And he would put it on a floppy disk. I don't know if you guys even remember a floppy disk. It held 1.2 megabytes of data. That's how we used to move everything around on computers back in the late 80s and early 90s. He would give the computer program to his friends on a floppy disk, but word got out, spread by word of mouth, and people from other institutions would say, can we get a copy of that program? And so he would post a disk to them, and people started using it. Well, when the internet came along, very quickly he figured out a way to distribute it. And what he did was he went to the National Institute of Health server, and he built this little web page right here. And to this day, it still looks like that. And I'm now going to switch out of the PowerPoint presentation. I'm going to call this image here, and we're going to take a look at the ImageJ website. Here it is. You guys are so used to seeing, super flashy websites these days. This is such an old-timey looking thing. Razbad created this in the early 1990s, when the program was still called NIH Image, by the way. So one of the changes that's happened over the years is that the name of the program has changed. The J stands for Java, and that's because very early on he realized that if people could program their own little subroutines, subprograms in Java, they could add functionality to ImageJ. So the original ImageJ program would open an image, it would allow you to set the calibration, set the scale, and it would make measurements like length and width and area. It would also let you measure brightness in an image, because that's the other important things that comes out of image analysis, how bright or dark is a region. And of course, that's what we're talking about when we're talking about analyzing the images that you're making with a fluorescence microscope system. So that's it. It's that basic. There's the program, and then there are a few links down here that you can click on. I noticed that they've taken off the little counter that they used to have down here that showed how many millions of times the program had been downloaded. Maybe you have to go to the download page to see that. It was up in the millions and millions. People use this program all around the world. And if you download the basic version of ImageJ, it comes with the sort of fundamental measurements built into it. And by the way, it looks really basic. It's a little tiny control window that opens up, and it really doesn't look like it's going to do much at all. Now, you can read about features. There's documentation that tells you about the program and things like that. But there's a link called plugins. I'm just going to go to that. We'll follow that link. It has a table of contents at the top of it. People started writing their little Java programs to do specialized image analysis techniques, things that they wanted to do. And when they got them working on their own data, he encouraged people to then upload their plugins to his website. And people are still doing that to this day. In fact, the number of plugins on the ImageJ website has increased almost exponentially over the last couple of decades. So when I first started using ImageJ, you could find 20 or 30 different plugins in this list down below here. Look what happens when I start scrolling these days. These are all things that the program can do. These are all little sub-programs. And it's ridiculous. It just keeps going. There are hundreds and hundreds and hundreds of them in there. You can go down a rabbit hole. You can spend a whole entire day playing around, looking at all of the different types of analyses that people have come up with. So this deceptively simple little web page here as a landing page hides this amazing plugin set here. And by the way, today you would call plugins apps. So these are in-app apps in a sense. You can get this program to do anything you want to analyze your images. So if I want to track moving particles, I can get a plugin for ImageJ that will focus on those little particles, outline them, and it will track them in time-lapse movies and tell me how fast they're moving and what direction they're moving and all sorts of things like that. It's just countless. I could do like an entire lecture on just the things that ImageJ can do. So there. That's that. The only change, by the way, I just noticed, the only change that's happened on this web page here in recent memory is the addition of this run ImageJ in a browser thing, which has the new tag beside it. So that's new. Nowadays, you don't even need to download the program anymore. Click on the browser link, and you can do the basic image analysis right on your web browser. So here's an example of one of those image analysis techniques, one of the thousands of techniques that you could do. This image here is part of an epidermal cell from the leaf of a tobacco plant. Endoplasmic reticulum marked with green fluorescent protein, Golgi bodies marked with red fluorescent protein. I call that the Scottish Terrier image. I don't know if you can see the dog, his ears, his nose, his beard, his tail, his legs. Front legs are bent up here. So in the Scottish Terrier image, we've got lots of stuff marked that we can analyze. It's a single frame from a time-lapse movie. And when the movie plays, you can see the endoplasmic reticulum and the Golgi bodies moving around. Well, I use a plugin in ImageJ to track the Golgi bodies. So those little red dots move around. I track them. You can see that it puts a little dot for every single frame in the movie. And there's little arrows showing which direction a Golgi body is moving at any time. And we can measure how far they move and how fast they move. And then we can render that graphically using various different techniques. Here, I was looking to see if there was any directionality within the cell. And the answer was no. They seem to move relatively random. They go in every such direction, and they seem to move over different distances at a relatively consistent rate of speed. So that's just one little finding of all of the image analysis things that I've done over the years. Anytime you collect data from a microscope system, you will analyze it in some way, shape, or form. And finally, to finish this lecture on advanced imaging, I've got to mention electron microscopy. I'm not going to go into big detail about electron microscopy, but I want you to understand that it is a very useful technique. Obviously, in cell biology, look at the kind of images that we get out of a transmission electron microscope. There are the Golgi bodies. Look at the beautiful detail of the membranes, the membrane stacks that we call cisterna, the little vesicles that bud off from the Golgi bodies and deliver proteins around in the cell to where they need to go. So almost our entire understanding of the structure of things at the organelle level down in cells is because of transmission electron microscopy over the years. So back at the start, I told you that electron microscopy produces images with higher magnification and resolution than is possible by light microscopy. But there was a big caveat. There was a big but. Cells must be killed by fixation using chemicals prior to imaging. So with electron microscopy, we can't look at living cells. So there was this trade-off. Beautiful high resolution, no ability to look at how proteins are moving. So we can't do those kind of experiments like the FRAP experiments or the FRET experiments. But we can see the wonder of nature. Transmission electron microscopy is made for looking at very thin sections of tissues that have been stained, so that you can see the things inside of cells. That's probably more pertinent to cell biology than is scanning electron microscopy. Scanning electron microscopy makes images of the surfaces of structures. So you can turn the scanning electron microscope up to very high magnification, and you can look at the surfaces of things like cells and pollen grains and what have you, but very often we use it at slightly lower magnification to look at the whole three-dimensional structure of something like, I don't even know what this thing is, but it's obviously the mouth parts of some worm or feeding organism. And by the way, images that come out of electron microscopes are always black and white. When you see these amazing colored images, they have been colored using software. People that do scanning electron microscopy and like to produce beautiful images become real masters of the Photoshop techniques. So this would be a black and white image that's formed here. I'll show you a couple of other SEM images as we go along. Anyway, let's talk about transmission electron microscopy for looking at very thin sections of cells. There's another beautiful thin section of a cell. Here we have the Golgi apparatus here, blebbing into these great big vesicles. And you can go out and you can see the plasma membrane around the outside of the cell here. You can see all sorts of little dots within the cell, which may be ribosomes and they may be other structures. This structure here is a mitochondrion, for example. I can see all sorts of things going on just in that one picture. And that's at comparatively low magnification, by the way. Scanning electron microscopy. There's a mosquito standing on some, I believe they're salt crystals, if I remember the caption for that picture. So absolutely beautiful to see the details of insect mouth parts and wings and eyes and things like that. Bacteriophage. That's a T4 bacteriophage that would have been sitting on the surface of an E. coli. Just a beautiful, beautiful scanning electron micrograph. Here we've burst open a cell that's at metaphase. You looked at metaphase chromosomes like this in one of the practicals in cell biology and genetics in first year. Here is that same sort of thing looked at with a scanning electron microscope. We can beautifully see the structure of the chromosomes, again, still at fairly low magnification. So how does this work? Electrons interact with heavy metals which have been added to sections for transmission electron microscopy, or which have been used to coat the specimen for scanning electron microscopy. In a light microscope, just by way of review, you've got a lamp or a laser, if it's a confocal microscope, which fires a light which is focused by lenses, glass lenses in light microscopy, onto the specimen, which is at this level here. The image of the specimen is then refocused either to the eyepiece or to the camera or detector, which forms the image on the computer screen. Bottom line is you're using photons, which are focused by glass in light microscopy. In electron microscopy, we're using electrons, which have a much, much shorter wavelength than photons, and that means that they can resolve things with much higher power and produce much higher magnifications. Electrons, though, won't go through glass. They just don't have the energy to do it. So this is a big column that the electrons fire through here to go through the specimen, and the column is completely evacuated, by the way. All of the air is taken out of it because even air molecules will stop the electrons, and you focus the electrons using strong magnets. So the things that are called condenser lenses are glass in light microscopy, but magnetic rings inside the column of a transmission electron microscope. Electrons are emitted from an electron emitter. They're focused onto the specimen, refocused down onto a detector, which is indicated in this picture as a fluorescent viewing screen. And that's how we saw the image in the old days. You would sit in the electron microscope room and you would have a phosphor screen, and you would put your eye up to the binoculars and you would see the image forming as a sort of a greeny image on the phosphor screen, and then you would take a photograph of it onto film. Well, nowadays, of course, the image is formed onto a charge-coupled device, which is a type of detector, and it appears on your computer screen right away. So that's transmission electron microscopy. Transmission implies that the electrons go right through your specimen, which is right there. Look what happens in scanning electron microscopy. You've got an electron gun beam that's focused onto the specimen, and you've got a coil that scans the electrons over the surface of the specimen down here. When the electrons hit the specimen, they don't go through it like they did in transmission electron microscopy. They bounce off of it. So electrons bounce off of the specimen and they go into a detector, and that's what forms the image. They can also, the energy in this beam here can also generate new electrons which are shed from the surface of the specimen, and those also go into a different detector, and those can also be used to form the beautiful, beautiful three-dimensional image. So transmission through the specimen, thin sample, scanning, electrons hit the surface and bounce off, a relatively three-dimensional specimen. Entire textbooks could be written on all of the preparative techniques for electron microscopy. It takes a long time. In the scale of things, in the grand scale of things, if you had some cells Or even better, if you had a seedling of a plant that was expressing green fluorescent protein, you could pluck the plant off of the little petri dish where you had germinated the seed, put it in water on a microscope slide, put it on the confocal microscope, and be making an image of the green fluorescence within the living cells in about 2 minutes. That's all it takes. If you wanted to make images of what was going on inside of the cells of that plant using a transmission electron microscope, it would take you at least 2 weeks to prepare the specimen properly for imaging. If you wanted to image the plant, the three-dimensional little seedling, with a scanning electron microscope, it would still take at least a couple of days to do the preparation to get it ready for imaging with the scanning electron microscope. So, in a nutshell, it takes a long time to prepare specimens, especially for transmission electron microscopy. The people that do it are people that have been training for years. So what they usually do is they get into a lab doing a project or something like that, and they learn some of the basic techniques. And as you learn the basic techniques all the way through, you get really, really good at it. So in our Center for Bioimaging at Brookes, we have a lab manager whose name is Flavia, and Flavia is an expert on electron microscopy, all techniques, and she employs various people that know how to do more or less the preparative techniques, and all of them are on a learning trajectory. So with electron microscopy, it's not easy to teach it as part of a single practical or something like that in a module. We do, though, in most years when we can get in and get our hands on the microscope, unlike in this COVID year, we do get you in. We have samples that are pre-prepared, so at least we let you run the microscope and make images of cells. Anyway, that's skipping over a lot of the steps that are indicated here. I'm not going to talk about all of these in huge detail, but you need to take your tissue that you're going to want to image, whether it's cells, whether it's a piece of tissue like liver or kidney that you've cut out from an organism. You dissect a little wee bit of the tissue that you want to look at, and you put it into fixative. Fixatives are chemicals that cross-link proteins. They join all of the proteins together so that the things inside of the cells remain relatively in place like they were in life. Now, you've all heard of formaldehyde, which is the famous fixative that even back in Darwin's time, they were pickling things in formaldehyde. You would take whole tissues and organisms and put them in a jar of formaldehyde. Well, formaldehyde is a fixative. It's not a good one for electron microscopy because although it preserves the structure of an organism, it doesn't preserve the structure down inside of cells very well. Things that are inside of cells all coagulate. They don't look very nice when you look at them by the TEM, but if you use the big brother fixative, which is called glutaraldehyde, you can cross-link all the proteins together and preserve the cells so that they're ready to go through the next steps of the process. So you do your fixation. Fixation is done with aqueous chemicals. So this would be glutaraldehyde solution in water. Ultimately, you're going to want your tissue to be embedded down here in plastic or resins. And most of those things aren't miscible with water. So the next step after fixation is to remove the water from your sample. You remove the water by doing this dehydration process very gradually. You start by transferring your specimen from the fixative. You wash it with water. Then you move it from water into, for example, 5% ethanol. Then 10%, then 30%, then 50, 70, 95% ethanol, and finally 100% ethanol. The reason you go so gradually is if you just took your water-filled piece of tissue and you plunked it directly into 100% ethanol, it would cause it to go like this and shrink and dehydrate horribly. So you have to go slowly and gently at it. So it takes time and it takes a bit of zen. People that do electron microscopy well are people that are very organized, that know all of the steps and really take their time doing it. So that's the dehydration process. Usually, in fact, we want to go beyond the ethanol or the acetone that we've used in the dehydration series. We want to get the tissue into propylene oxide, which has a very high vapor pressure. It's a solvent. Once you've got your tissues into propylene oxide, then you want to embed them, and you use liquid plastic or resin to do this. You take the tissues out of the propylene oxide, put them into the liquid resin, and then you let the resin cure and set solid. Now the point about using the liquid resin is, of course, to support the outside of the tissue so that you can section it, but even better, you've also got to get it to go into the cells. So when you look down at a specimen, a picture, an image rather, that's been made with an electron microscope, it's not that the 3D cell has been surrounded by plastic, it's that the liquid plastic monomer has penetrated all the way into the inside of the cells. Everything is completely impregnated with liquid plastic or resin, and then that is hardened. So now you've got a block that contains your sample. You use, and this is fun, you get really good at this, you use a little jeweler's saw and you cut out the little piece of plastic block that has your piece of tissue in it, and then you get it ready for sectioning. And I've illustrated this over here. We've got machines called microtomes. You attach your resin block, which has your little bit of tissue in it here, attach it to the microtome. The microtome works automatically for electron microscopy. It's not like microtomes that you might have seen for sectioning wax, where you rotate a big handle around and the specimen goes up and down over top of the knife blade and it cuts thick sections. The sections for electron microscopy are called ultra-thin sections. So the machine works automatically. It moves the specimen like this, up and down over top of a blade. You can use a piece of broken glass, as your knife blade, and there's a real skill at making glass blades, so-called, that are nice and sharp, wickedly sharp. But something that's even sharper and lasts longer than a glass blade is a diamond blade. So we're using diamonds here in this electron microscopy step. The diamond knife blade, by the way, can cost thousands of pounds. This specimen moves by the diamond knife blade in little teeny tiny thin sections. There they are there. There's 4 of them indicated. Float off on the water or the liquid that you have in this little trough on the outside of the knife blade. Once your little section is floating there on the solution, you can pick it up. So you go in with one of these little metal microscope grids. The reason they're little metal grids like this is that the metal part, usually copper, supports the section that you've cut. That's 3 sections there that are stuck together in a ribbon. But of course, this mesh is open here so that the electrons can go through. So if you look at the way these sections are sitting, the electrons can go blasting right through the middle. Electrons, by the way, will bounce off the grid bars in the mesh, but that doesn't matter. You can still see what's going on inside of your cell by looking down through a part that isn't lying over top of a grid bar. So we've done this. We've embedded in resin, we've sectioned, we've picked the sections up, and even that is an art form, how you pick the section up onto the grid. Now we're ready to stain them, because of course the tissue inside of cells isn't going to stop electrons at all. These sections are so thin, there's the thickness right there, 50 to 100 nanometers, unimaginably thin. Electrons will go whizzing right through that, which is what we want. We want the electrons to go through the resin where there's nothing interesting in the tissue. I'll show you a picture to illustrate what I mean on the next slide. So electrons will go right through there. We need to somehow stain the good stuff inside of the cells. We've got to stain the membranes. How else would we see the mitochondria, the ER, the nuclear envelope, the ribosomes, and all those sorts of things? So we stain by adding heavy metals like lead and uranium to the cut sections. We use lead and uranium chemicals that actually bind into membranes, and that's how we stain biological tissues. And then, once you've got your sections on the grid and they're stained, you put the grid into the holder in the electron microscope and you image it. Electrons pass through the sections where there's no heavy metal and form white in the image. Electrons are deflected by metals attached to tissue components like membranes and form black in the image. Here we go. Here is my biological specimen on a grid. The grid bars would be here supporting this tissue. Electrons are fired from up above out of the electron gun. They're focused onto the specimen. Where they hit a heavy metal atom that's in a membrane, they deflect away. They never make it to the detector, and the detector therefore stays dark. and it forms black in the image. But where an electron doesn't hit a heavy metal atom, it goes right through the specimen, it hits the detector, which therefore forms bright or white. So you get a black and white image, just like the one that's illustrated here of this beautiful Golgi body. Where there were membranes, the electrons didn't get through and we got black in the image. Where there were no membranes or real biological material, the electrons got through and we got white in the image. And you see the beautiful, beautiful details of the biological specimen. So that is the world's fastest tour of how you do transmission electron microscopy. Basic specimen preparation for scanning electron microscopy is completely different. There are, in theory, there are some commonalities, but you start by fixing the specimen usually. You could use formaldehyde in this case. You don't need to cross-link things inside of cells because you're not really looking at the inside of cells. You're just looking at the three-dimensional structure of whatever your biological specimen is. So you do fixation, then you dehydrate up to ethanol, just like you did for transmission electron microscopy. Maybe you can move a little bit more quickly through the process in this case here. Then we've got a technique called critical point drying. What's important is to get rid of all of the liquid of any type that's inside of your specimen. Well, you've got ethanol inside of the specimen. We can't put ethanol, a specimen containing ethanol, into the vacuum column inside of the microscope. So we have to dry all of the liquid off. Well, what happens to a biological specimen when you dry all of the liquid off of it? You can go look in your fridge right now, look in the vegetable crisper, find some vegetables that you've left sitting in there for 6 or 7 months because you don't eat vegetables. What happens to them? They shrink and they dry. Biological things dry out. Well, that's never going to work. If we just dried the specimen, it would lose its beautiful three-dimensional shape and just dry to a little husk. It wouldn't give us any information. So we do this technique called critical point drying. I won't explain the physics of this, but what it does is it drives the moisture, the liquid, out of the specimen by turning it to gas immediately so that your specimen doesn't shrink at all. You get these little specimens that come out of the critical point dryer that will just blow away from your workbench if you just breathe on them. They're so light, but they're still beautiful, beautiful, three-dimensionally preserved. That wasn't an English sentence. They are beautifully preserved three-dimensionally. That's what I'm trying to say. Then you want to mount the specimen onto what's called a mounting stub so that you can put it into the electron microscope. And there's an art here as well. If you've got like that picture of the mosquito we were looking at, you don't want your bug to be lying down on its side necessarily. Whoever made that beautiful image of the mosquito on the salt crystals spent a whole lot of time, put the salt crystals, they used their very, very fine forceps, and they got the mosquito standing up on the salt crystals. They probably did all of that while looking through a dissecting microscope. You get it on there, get it glued in place, and it's almost ready to be looked at with the microscope. There's something that we've missed out though. Do you remember that I told you you need a layer of heavy metal atoms on the surface of the structure so that the electrons will bounce off into the detector? So you coat the entire specimen with a heavy metal. And the heavy metal that we use in scanning electron microscopy is usually gold. It can be platinum, something like that. It's usually one of the precious heavy metals. And people are amazed when they hear that we coat things with gold. It's a super thin layer, 1 atom to 2 atoms thick maybe, so it's worth a couple of pence worth of gold, but we use real gold foil in this machine that's called a sputter coater, and we evaporate the very thin layer of gold onto the surface of the specimen. When the specimens come out of the sputter coater, they're gorgeous. They're on the little stub. There's your mosquito, for example, standing on the stub, and it's completely gold and shiny. It looks like a little piece of jewelry. Then it goes into the specimen holder of the microscope. You blast the electrons down onto the specimen. The gold deflects them into the detector, and the beautiful, beautiful three-dimensional image is produced. So transmission electron microscopes, this is the one that we have at Oxford Brookes. You can get the scale of it because you can see the computer monitor there. Here's where you would sit. That was the viewing screen I was talking about. You could look through there, but these days people tend to just look at the computer screen here. The specimen goes right in here. The electrons are generated up here, fire through the specimen onto the detector or the camera, which is down below, and you see your image form here. And of course, there's quite a lot to learn about running the microscope when you're imaging. You can of course adjust zoom and things like that. You can adjust the fineness with which you image the specimen, but there are lots of other things. Somebody that's an expert using electron microscopes will learn to adjust those condenser apertures and things like that so that the electron beam is perfectly centered and they get the best resolution possible. I just like this picture here. This microscope is made by the same company, Joel. This is the Joel JEM 1400 flash. Here's a Joel microscope here. It's a high voltage million volt transmission electron microscope. And if you want to understand the scale of that picture, look for the chair where the operator sits. It's right there. Look at the size of that. The electrons accelerate from up here down through a little specimen. The higher accelerating voltage gives you the possibility of higher resolution and greater magnification. It means that they're able to go right down and image macromolecules, tiny, tiny parts of DNA. Look at the structure of the individual nucleotide bases where they join with each other. Here's just a couple of tricks you can do, though, with transmission electron microscopy. You can do a negative stain. This is a bacterial cell here, and all the little things that you see around the outside of it are the T4 bacteriophages. You can do shadow casting, in this case, to see the nucleic acids, which are unwound here in interphase. This is the little scanning electron microscope that we have at Brookes. It's a tiny one, but it's a good one. Does a good job. We put lots and lots of project students through this. Third-year project students use this microscope quite a lot in a normal year. Lovely little thing. It's made by Hitachi. The one thing I am guilty of saying quite often is that the transmission electron microscope is much more useful for cell biologists, and I still sort of maintain that. If you want to see what's going on down inside of cells, you want to use transmission electron microscopy. But the fact that the SEM shows you the three-dimensional structure of things is working for a lot of the biologists in our department. People that study the blood-borne parasite called a trypanosome make all sorts of mutations of this single-celled organism that affect the body shape, and they're doing lots and lots of scanning electron microscopy to look at the shape of the organism once they mutate gene X or gene Y. The people that study eye development in Drosophila, and I don't know if I've said this to you guys or not, why would you want to study eye development in an insect? Well, because the genes that control the fruit fly's eye development have homologs in humans. So if you're concerned with studying human eye development and studying the reasons for blindness, why not go back and use a model organism like Drosophila? And one of our groups, or a couple of our groups now, are looking at Drosophila eye development and making absolutely gorgeous images using scanning electron microscopy. So there, I just finished telling you that a couple things, that scanning microscopy isn't as useful for cell biologists, and that the scanning electron microscope that we have at Brookes is quite a small one, but I've been holding something in reserve. Back at the start, the very start of the week 1 lecture, when I told you about the difference between light and electron microscopy, I said that confocal microscopy lets us make optical sections down through a specimen and look at them as three-dimensional reconstructions, which is really, really helpful, really, really beautiful. And I said we can't do that with electron microscopy because a transmission electron microscope is made to look through a single, very, very thin section. Of course, people can cut those very thin sections so that they preserve them in order on the grid, and they can make an image of each section one after the other, and they can try and stack them up with software, and people have spent a lot of time doing that. Other techniques have been developed for looking at a three-dimensional sample with transmission electron microscopy. There's a technique called tomography, for example, which I won't talk about right at the moment, but there's another solution. I told you a while ago that the scanning electron microscope just makes images of the surface of a specimen. But if you have a big, very capable scanning electron microscope, and you embed some cells in resin, and then you section the top off the resin, and you stain the surface of the block so that your membranes are stained with lead and uranium and things like that, and then you make an image using an SEM, it will show you an image of, for example, the nucleus, the endoplasmic reticulum, what have you, and the image looks very much like the image that we would get from a transmission electron microscope. This takes a very capable SEM, a big one. Zeiss invented a microscope. It's more than 10 years ago now, but Oxford Brookes has one. It's called the Merlin Field Emission Scanning Electron Microscope. And it's a big one. It's a very capable scanning electron microscope. In fact, when Oxford Brookes first bought this microscope, we needed a separate room to house it, and it went into what was my research lab. I had to go work in a teaching lab for my research for a while because we had no other room for the microscope. Well, now in our new bioimaging center, this thing takes pride of place. It has its own whole room, and it's an absolutely monstrous and beautiful machine. It can make an image of the surface of that block. But I hear you asking, how, though, does that make a 3D image? Well, it's got a trick. The confocal microscope did optical sections, and I was all proud of telling you that that was completely non-invasive. You could do that on a living sample. An electron microscope, even this big Merlin thing, can't do non-invasive 3D imaging, but it has a trick. What they've done is they've put one of those sectioning microtomes into the microscope here where the sample is. The microscope blasts its electrons onto the surface of the block, and it makes a picture of the surface, which is the equivalent of a very thin section. Then the microtome blade cuts away a very thin section right off the top of the block, and the microscope makes another picture. Microtome, picture. Microtome, picture. The microscope can keep doing that all night long. It can cut hundreds and hundreds of sections through a block and make a picture after each section is cut so that you can then take all of those pictures into your big, powerful image analysis software package and generate 3D views of what's going on inside of cells. Well, we've done some cool stuff with this at Brookes. Professor Chris Hawes looked at plant cells, and he did the three-dimensional imaging thing with this system, stacked all of and I think there were probably 300 images that went into making the picture of this one cell here. The nucleus is in yellow, endoplasmic reticulum is in the turquoise color, Golgi bodies, I believe, in green, and mitochondria in purple. What I'm showing you here, though, in the PowerPoint presentation is this incredibly low resolution, low magnification view of this thing. After the microscope has done its job, if you've got this on your great big computer with all of the individual images, you can zoom right up on this. You can fly right down into the cell and you can see all of the details of how all of these organelles interact with each other. It's absolutely beautiful. I've got an example here of 3D sectioning using the Merlin system. Let me just show you something on the previous slide. This is a trypanosome inside the blood of someone who has a trypanosome disease, like sleeping sickness, malaria, what have you. It's going to get the movie going and then stop. We're looking at the surface of a block imaged with the SEM. And it's really kind of hard to tell looking at that blob right there what that is. Here's another blob here, another blob here. Are those all individual blobs? Let's play the movie a little bit more. More individual blobs. It seems like we're looking at a bunch of individual cells, doesn't it? Let's play it some more. What emerges? What emerges is a trypanosome. Those individual sections, cut through here, for example, you would have seen a little blob. Cut through here, another blob. Down here you would have seen a little bit, and over here another little bit, and over here another little bit, because of the highly curving nature of this little creature. So this microscope does the 3D sectioning and then the software image analysis. puts all of the sections together and makes the 3D representation of the trypanosome at very high resolution, very high detail. This is a surface view, which somebody has colored to indicate different structures. That's nice, but watch, we can go further. We can start to strip away the surface, and we can see the details of what's going on inside the cell. And if you look at that little picture that's moving back and forth there, let's go back, it looks just like a transmission electron micrograph. You can see all of the individual organelles, and then those organelles can be colored in so that we can see exactly how they relate to each other. So really, really super detailed view of the trypanosome, its morphology and its anatomy, the interior structure of all of its organelles. And that's really helping in the fight against those trypanosome-borne diseases. Last slide. And I just want to summarize. This was light versus electron microscopy. I told you about how amazing confocal microscopy is, the ability to look at living cells in a completely non-invasive way. And I showed you that movie of the little Golgi bodies moving around in a plant cell. I talked to you about fluorescent proteins, and of course that's how we label the different organelles that are inside of cells. So that's absolutely beautiful if you want to do the sort of experimentation that lets you measure how proteins move around or how organelles move around with each other. And I continued the story all the way through electron microscopy, and I told you about this amazing new electron microscope that will let you do three-dimensional imaging of the organelles inside of cells. And I want to show you this movie here. This would be best if you had the 3D, you know, the blue-red glasses that let you see the 3D image, because this has been rendered in 3D. If you've got some of those glasses, put them on and watch this. But if you don't have them, I'm sorry, you'll just have to imagine for now. The Golgi bodies, by the way, are these organelles here that have all the vesicles blebbing out from them. Let's watch the movie. To me, there's just a wow factor in that. That's absolutely gorgeous. I mean, it looks like pin sharp, and it feels like you're down inside the cell if you're wearing the 3D glasses and can really see it. So we're seeing electron microscopy resolution of 3D reconstructed Golgi bodies there in fixed and killed cells, and we're seeing the little Golgi bodies doing the stuff of life here in the confocal image. Okay, thanks for listening, everyone. Bye-bye.